thank you all for coming. Thanks to Michael Teskey for inviting me and his colleagues for arranging this event. It's an honor to be here. So Michael asked me to talk about what aspects of Reed shaped my life. He asked me just two things. One, to talk about Reed and my career, and two, to keep it short. So here I go. So I wanted to mention just four things. So the first one was, I think, Reed really taught me the importance of defining a question. And the reason I say that is I have a very vivid memory of being in my plant physiology class of Professor Helen Stafford, and her explaining to us that there were fungi that can actually colonize plant roots and that they have to communicate with the plant in order to do that. And I was astounded by that. And at the time, I wanted to know how plants and microbes communicated and why. And because at Reed you do a senior thesis, I was able to think about that question for a whole year. For my thesis, I looked at mycorrhizal fungi recolonization of plants after Mount St. Helens had blown up. I did spend a lot of time thinking about these questions. How do they communicate when they can't speak, they can't write, all the normal forms of communication that I'm used to? I ended up doing a fellowship in Sweden to study mycorrhizal fungi. There was a professor I had read about when I was a student at Reed who studied this very question. And he was one of the few people that had really advanced knowledge in understanding what sort of chemicals the mycorrhizal fungi produce. So I went to Sweden, I spent a year there, I futzed around in the lab, and I realized this is a really bad system to work on. So the mycorrhizal fungi have to be grown together with the host plant, and you can't do any genetics, and it was really frustrating, but I was still very drawn to this question. So I ended up in graduate school working on bacteria. They're single-celled organism, very simple to genetically manipulate. And then I went on in my career at UC Davis to look at interactions with the rice plant. So in 1995, we answered part of that question. My laboratory discovered a single gene in the rice plant that makes the plant completely resistant to a bacterial pathogen. So that was part of the question. This gene made what we think of as a receptor, sits on the outside of the membrane, and it recognizes something in the bacteria, and upon that recognition, it sets up a whole defense response. So these so-called resistance genes are in everything that you eat. Farmers really rely on these genes to combat pests. So that was a very exciting time to really answer that part of the question, but we still needed to understand, well, what is it that this receptor, this alarm system, is recognizing, and why? Why would a bacteria produce some sort of molecule that the plant recognizes? So just in the last few months, members of my laboratory have now isolated a molecule in the bacteria that is important for the signaling. And it turns out what we believe is the molecule actually is meant for the bacteria to communicate to each other, to tell the other single-celled organisms, let's get together and let's turn on our attack mechanisms. And as they're getting ready to attack, the plant now has intercepted this communication. And that sort of signaling mechanism is really what explains this particular disease interaction, and it's also been found in many other plant pathogen interactions, and similar mechanisms have now been found in animals. So that just gives you an example of the importance of defining a question. If I had not had that opportunity at Reed to think about that question, I wouldn't have moved along this very exciting and rewarding career path. So the second point I wanted to make is that I really think I learned at Reed that if you want something, you need to be persistent. And I was very shy. When I came to Reed, there were all these incredibly talented students, and I had not somehow I had not really known that people so young could be so talented. And one of the most inspiring aspects of READ was really getting to know the other students, to see what they could do, to see the power that they had to harness their skills to move forward. And that was very influential to me. I thought, well, maybe I could try that. And I really appreciate that. And I think one reason, so Philip mentioned this book I wrote with my husband, and we sort of decided to do this book before we knew how much effort it would take. My husband and I, he's an organic farmer and I'm a geneticist, so we're often asked about organic farming and genetic engineering, and will organic farming be enough to feed the world, and will genetic engineering contribute to sustainable agriculture, or will it create some ecosystem issues? So we really felt that we had a voice that was somewhat unique, and we decided to combine our skills and write this book. So you can imagine there's a lot of challenges writing a book with your husband, and he's a farmer and I'm a scientist, and we've never written for the general public before. So I think that persistence that I learned at READ really paid off, because we did finish the book. And I do think, though, that if READ had had a creative writing program, it would be a much better book. So the other aspect I learned at READ is, if you don't want to do something, well, just don't do it. And that's a little more difficult than you realize, because I think a lot of us, especially some of us that are academically inclined, we get started on a track. And that's what we're supposed to do, and sort of the system encourages you to continue. And I really loved genetics at READ. Peter Russell was my professor, and I just fell completely in love with genetics. So I went to work in the summer in the laboratory of Larry Rubens, who was a wonderful mentor, a very kind person, and was very encouraging to me. The problem was, I had to cut up frogs, sew them up, and sometimes the frogs would die. And I didn't really confront the fact that I hated it. And I actually went on after college to go work in a laboratory at Stanford doing the same kind of thing. And it was really a disaster. I would have nightmares about the frogs. My employer wasn't very happy with me. And I finally decided I was not going to do that. And I ended up working in a plant genetics lab. And it was fabulous, because plants have genes, but they don't have blood. It was just perfect for me. And I know that if I had tried to continue along that path of animal genetics, I never would have had the opportunity to be a professor in a profession that I truly do love. I wanted to just end on a point. Actually, Philip, you already mentioned this idea of discussion. Discussion is important, and it's something that I think we take for granted. Many of us that have been educated at READ, or perhaps some other very nice colleges, and of course READ really fosters this discussion. You have, from your first week, you're with a group of people that are very different from you, very different backgrounds, very different interests. This idea of getting together with diverse viewpoints and just talking about things, and really using critical reasoning and, I would say, science-based decision-making is really very important. So I really like that I had that opportunity to learn to have discussions with people very much unlike, that are very different from me, and to enjoy those discussions, because we have some very challenging situations here in our home on this planet, not only global warming, but feeding 9.2 billion people with less food and less water. And so we need many different viewpoints. So really, those are the 4 points I want to make.